All right. Well, it's a few minutes after, so we'll go ahead and get started. It is really my pleasure to introduce Dr. Rohlman. She's joining us from Oregon State University, where she is in the College of Health, and she's actually a leader of the Community Engagement Core for their Superfund research program. I met Dr. Rohlman a few years ago. We actually collaborated on a small pilot project looking at VOCs and preterm birth and really uh, wanting to look at ways to report back research results. And I'm really excited that she's going to be presenting hopefully some of those findings in her talk today, titled Report Back of Research Results in Pregnancy Cohorts in Two Cities. So thank you so much again, Diana, for joining us. I know, like you said, it's early over there, so hopefully you've had your coffee this morning. With that, I will turn it over to you.
Thank you. You know, the funny thing is, I'm actually from Seattle, which is like Starbucks, all the coffee. I don't drink coffee at all. It's never worked well. But yeah, no, thank you so much for having me. So yeah, Jen introduced the title of my talk. I feel like this might be a slightly different talk. I always kind of view it more as like scientific storytelling. But to preface, what I'm hoping to do today is talk about like what comes after the data collection. So a great deal of my work has been doing exposure studies and then taking that information and giving it back to study participants with the idea of, you know, this is their information, they may find value in it. So I'm going to walk you through the process of two different studies, one in New York and then the pilot study that Jen was just talking about in Detroit. So I always like to start, you know, my students are always like, oh, what's your day-to-day life like? And I like to pretend that my day-to-day life is out in the field collecting samples, and that's a part of it. The majority of my work, of course, is sitting in front of a computer, but I always like to just sort of preface that, you know, a lot of my research really is on identifying and characterizing environmental exposures, and we do this with a variety of sampling methods and materials. So a lot of it is using passive samplers. I have done work around butter clams. That was actually with an Indigenous community concerned about exposures through their food. We've worked with asthmatics. We've done work following Hurricane Harvey and chemical exposures. I've even set up an air sampler in a football stadium during a wildfire. So various different methods that we use, but what we are trying to do is collect this exposure information and then translate it and return it back to the communities we're working with. So I recognize this term may be very familiar to several of you, so apologies if you already know this, but I'll be talking about report back of research results, or the really clunky acronym of RBRR. So the way I look at it is it's the return of research results. This could be individual level. So, you know, here are the results in this individual to de-identified aggregate summaries of results. I think it's been pretty well established that there is an ethical obligation to return results. With that said, there's not a lot of guidelines for how to conduct a report back of research results, particularly in pregnancy cohorts. So I wanted to just talk about how we approached this in two separate pregnancy cohorts. I should also mention, I don't know how you run these seminars. I'm very used to being interrupted with questions. So if people would like to do that, you are more than welcome, or you can hold them until the end, but I'll let you all sort of decide how you want to do that. So the two cohorts I'm going to talk about, I'll give a very brief overview. The first was in New York City, New York. So I worked with Dr. Julie Erbsman, who directs the Fair Start cohort. So this is the Columbia University Children's Environmental Health Center. So we were looking at pregnant individuals. To date, we've reported back individual results to 190. The cohort is actually over 300, so this is still ongoing. Here, we were looking at prenatal exposure with the outcomes of childhood asthma and respiratory health. This cohort is about 92% Latina. And in the study design, we used a couple different approaches. So we used a silicone wristband. Individuals wore that for 48 hours in their third trimester. They also simultaneously wore an active air pump and then filled out questionnaires across over about a year, we'd say. I'll talk about the silicone wristband a little bit more. Here, the chemicals of interest were 62 polycyclic aromatic hydrocarbons, so PAHs, formed from incomplete combustion. So in New York City, we were often looking at a lot of vehicle exhaust, industrial air pollution, those sorts of different sources of PAHs. I don't talk about this too much because I'm pretty sure everyone on the call is very familiar with Detroit, Michigan, but this was through the CLEAR study. This was a pilot project. So pregnant individuals, an N of 30, again, looking at prenatal exposure with the outcome of preterm birth. 72% Caucasian. Here, individuals wore a silicone wristband for 7 days while pregnant. In this particular study, we ended up looking for over 1,500 semi-volatile and volatile organic compounds. So I'm mentioning the chemicals mostly because that's what we reported back. I'm not in this talk going to share the actual exposure results, but more talk about the challenges we had when trying to return this amount of chemical data back to study participants. For a little bit additional context, we used the silicone wristband. So the silicone wristband was co-developed by Kim Anderson, who is also here at Oregon State University. And it is literally just a silicone wristband. They're purchased online. They do go, of course, through a cleaning process, a preparation process. But what we have found is that it's really quite useful as an exposure tool. So it's very lightweight, it's fairly unobtrusive. It doesn't require any electricity or internet. We can actually mail it in an airtight bag and then people can mail it back. So we don't have any storage requirements, right? There's no ice, there's no heat. One of the other benefits that we did not anticipate is that it comes in different colors. And that has surprisingly been one of the best ways to get people to agree to our studies. I'll just share a story. So at Oregon State, our colors are orange and black. At our rival, University of Oregon, their colors are yellow and green. We did not consider that. We did a study down in Eugene where University of Oregon is, and we brought orange wristbands and people refused to wear them. So we actually had to go back and make new wristbands in the color green. So that's just an amusing thing that we have found, something to consider. The other benefit to silicone wristbands is just the breadth of chemicals that we can detect. So it is going to be semi-volatile and volatile organic compounds. So anything in the particulate phase, so PM 2.5, mold, mildew, metals, we're not going to be able to pick that up. I'll also note we have made the decision that we will not look for pharmaceutical or recreational drugs, mostly just out of respect for participant privacy. But we can look for pesticides, polycyclic aromatic hydrocarbons, PCBs, dioxins, endocrine disruptors, quite a large suite of organic chemicals. So with that background on the silicone wristband, I want to then sort of transition into, that's how we collect the information. But once we have the data, right, what chemicals people were exposed to, at what levels, at what frequency, how do we give that information back to people? And so I like to start with the challenges because that's usually where we start. And we say, like, we've got all this information, what are we going to do with it? So there's a couple of different challenges. The first is just the breadth of chemicals that we reported back. So like I said, in New York, we looked at 62 polycyclic aromatic hydrocarbons. In Detroit, we looked at over 1,500. So I wanted to just sort of give a little bit of background on some of those challenges. So I did mention New York was 62. That was back in 2018. So since then, this method has grown. I'm showing the method for 63. The method is actually, I think, bumped up to 67 at this point. But we're looking for these 63 polycyclic aromatic hydrocarbons. In some benefits, there's 16 priority PAHs that the EPA has identified. And so all 16 are in that method. And so these are PAHs that were originally selected because they're commonly found at hazardous waste sites. We might have more information about their toxicity. With that said, while we do have more information about their toxicity, it's pretty limited. So if we look at that top graph in gray, these are going to be relative potency factors. So only 27 PAHs have relative potency factors, meaning they could potentially be assessed in a risk assessment. But when we look for other health data, it's pretty limited. So we don't even have much information for carcinogenicity. There's very few regulatory standards or health guidelines associated with these chemicals. So for example, while I might be able to, well, I can say phenanthrene is associated with respiratory health, right? We know that exposure to phenanthrene can irritate the respiratory system. We don't know at what level. So when we're giving this information back to people, we can say, yes, you were exposed to phenanthrene, but we don't know if it's going to cause adverse health outcomes for you. And that's a challenge. Then when we go to the 1500 method, this is also called the massive method. And I'll be honest, for the first year, I thought it was called the massive method just because it's a massive number of chemicals. It actually stands for multi-analyte screen version 1500, but sort of has the same outcome there. So when we looked at these chemicals, we ended up categorizing them just because there's no possible way that we can give, up to 1,500 individual graphs back to people. So we tried to categorize it into sort of bins that people would be more familiar with. We do run into some difficulty because some of these bins are actual chemical classifications based on the structure, right? Like pHs, PCBs, dioxins. Others are more use categories, so pesticides, personal care products, those are use categories. But then we also have categories based on function, like endocrine disruptors. On top of that, there's a lot of overlap. Pesticides can also be endocrine disruptors. Flame retardants can be endocrine disruptors. So these are not distinct categories, but quite a bit of overlap. So that's one of the challenges. The other challenges that we face can be participant size, both in very large cohorts as well as smaller cohorts. And then looking at these populations and realizing that they can be very diverse populations, they can have distinct environmental health concerns, they could come from underserved communities. And the way we're going to report back is going to reflect the community that we're working with. And then finally, I mentioned this already, but it is very difficult to provide comparison data. So we don't have many standards, and we're not currently able to use a lot of the NHANES, so like population-based data sets. I'll come back to why the comparison data is important. So thinking about those challenges, we also, of course, want to frame the way we do our report back within requests we get from the community. So I'm going to note that in both New York and Detroit, we were really fortunate that we had really engaged community liaisons as part of this process from the very beginning. So these were people that were either from the community that were study participants or had worked closely with this community for years. And just not being hyperbolic at all, they were a wealth of information. So we were able to then take their experiences or their lived experiences, community priorities, and build that into the way we thought about the data and the way that we then visualized the data. So here's where we're saying, like, yes, we need to report back scientific findings, but we also need to separate, like, what are we really interested in versus what is the community really interested in? And probably the best example I can give for that is, we had a study where we were really fascinated because we were finding a lot of 5-ring PAHs, which is not something we see a lot of. That was not the community priority, right? They weren't really interested if it was a 5-ring or a 4-ring PAH. They were more interested in where they were coming from and what it might mean for them. So that is where, yes, we need to describe the study rationale, but we have to revise our reports to be reflective of the community needs, and then put those results in the lived experience context wherever we can possibly do that. So with that sort of background, I will transition into the actual 2 cohorts that we worked with and our process that we had. So we were very lucky in both of these. We weren't starting from scratch. So we previously developed templates for our PAH and 1500 methods. We'd only tested them each in one community, and they had very different contexts. So our PAH report was developed for use in an Indigenous community. And then the 1500 template, we actually developed that post Hurricane Harvey in Texas. So we were testing this with communities recovering from Hurricane Harvey. In both of these cohorts, right, with New York, it was the first time we were reporting back to pregnancy cohorts. And so we wanted to make sure that we were being really sensitive to and approachable to what pregnant individuals would be facing and not adding on to the stress of what they might be exposed to in pregnancy. So the New York study was held first in 2018, but what I'm showing here is we actually have very similar processes in both. So we did develop our reports internally. We revised them with community liaisons. In New York, we actually did have the ability to disseminate a survey to assess changes in knowledge. Then in both New York and Detroit, we held focus groups to evaluate the reports and then did a final revision and then disseminated those reports. Like I said, we started with existing templates. We were also really heavily informed from expertise from Silent Spring Institute and then the Centers for Disease Control and Prevention. So these are some of the guidelines that we had as we drafted our reports. So that first guideline is a little more specific to the Detroit group because we had so many chemicals, but we did really focus on chemical groupings. We still showed individual chemicals, but within those groupings. We tried to include health information where we could. We were also very honest when, that health information just didn't exist. I think it's really important to talk to people about what's known versus what is not known and how studies like this can help address the not knowns. That's terrible grammar, my apologies. I've already mentioned the context. We do our utmost to write everything in 8th grade reading level and then set expectations. What are our limitations? What are our future studies? We try and talk about what risk means. As toxicologists, risk to us is a very quantitative concept, right? We can put a number to it. Most people don't think about risk that way, right? Risk is actually more about feelings. So can we talk about risk in terms of how it might make people feel? And then, of course, always providing resources so that if people aren't concerned, they can then take steps to reduce or prevent their exposure. So I'm going to jump right into New York. We were really lucky in New York that we actually got to do a pilot test. So in this particular cohort, there is now over 300 participants. But when we started reporting back, we worked with the very first group of participants, so 22 people. What was pretty interesting is they came to our focus group and they brought the kids that they had been pregnant with when we first started sampling. And those kids were now 5 years old. So this was the very first time that results had been reported back to this cohort, which is why we wanted to pilot test it, see what we could do, see how it was actually being received by this population. In this group, we actually gave back reports with their real data for the focus group. That's not something we've done since for a couple different reasons. But with this group, we built reports for 22 participants with their data. We gave them a survey, and then we held semi-structured focus groups in English and in Spanish. So the survey was made to be accessible on smartphones. This population does most of their work on smartphones. And then we held these focus groups. And so I'm showing the questions here, but what I want to sort of preface everything I'm going to show you next. When we did these focus groups, it really was designed to be an evaluation of the report, right? What worked, what didn't work, how do people like to view their data? But as we did this, we really started realizing the long-term consequences of report back. So I'll slowly move my way through that, but I did want to set the stage that these were designed for just evaluation, but we found that we could do quite a bit more. So since I've been talking about the report, I wanted to show you what we actually gave to this first group of 22 people. So we started with a cover letter, hindsight being 2020. Landscape orientation was a terrible idea, and it's just a page of text. So we probably should have known better, but that is why we test these things. We also did include infographics, so help people understand what pHs are, how they can reduce exposure to these pHs, but they ended up squeezed on one page. From there, we jumped into a table. This is priority pHs. So we said of the 16 pHs, which ones were detected in your wristband? From there, we then pulled out a couple different pHs of interest. So we did select these based off of the purpose of the overall study as well as community interest. But I'm not showing, and I'm not going to show throughout this report just because it's kind of overwhelming, but we do end every report with a table of every single chemical that was detected in that person's wristband. So we do have full data transparency with our participants. So this was the draft report, and then we ask for feedback on it. I am summarizing over 3 hours of focus groups and 22 surveys. So this is very distilled down. Most of it is probably not surprising, but we got a lot of comments on the clarity. So saying reorder elements of the report. Think about, if you're a participant, what is the way that you would want to see the data? So we did some reorganization. There was a request for more graphics, more use of color. They mentioned that our language was too academic. They were very polite. Graph literacy was still an issue. And then I'm not going to spend a lot of time on this because I'll come back to this at the end, but there was definitely requests for comparison data, right? Like they wanted to know what are our exposures being in New York City compared to like suburban Oregon or to other large cities or to rural areas. And then quite a bit of requests for mitigation strategies. So how can they reduce exposures, but also where exactly are these chemicals coming from? So taking all of this information, which I'm not showing here in the interest of time, I'm just showing the overview, we substantially revised the report. So we moved away from the landscape orientation. We went more into letter style. These are 8.5 by 11 pages. We did try to reduce text, but we also brought these take-home messages onto the front page. So this is sort of an overview of the data and our scientific interpretation of it. We did also provide links to learn more about PAHs. So we've got videos, we have infographics, we have fact sheets. We did include one infographic. This is the same one we had before, but it's now full size. And it was starting to follow the order that people said. They said, remind us of the study, which is the cover page. Then tell us more about polycyclic aromatic hydrocarbons, because those do not come up often in conversation. And then go into what you actually found in our results. So we'd previously shown a table like this. What was really interesting, the table we had before, we are just listing different sources of chemicals. So for example, benzoapyrene is found in cigarette smoke. So in the table, we had an X under cigarette smoke. But pretty unanimously, our participants were like, oh, there's an X. That means it's not found in cigarette smoke. So they suggested we lay this out like a product comparison review that you might see on Amazon, right? Every time you go to buy things on multiple online sites, it'll say like, here's what you're looking at. Does it have these features with check boxes? So we were able to do that. What I'm also really proud of with this table that you can't see is behind the scenes, this page of the report is fully automated and completely personalized. So every place you see a number, that is actually coded to pull that individual's data and then automate this page. So it will say how many pHs were found in this wristband. It will only show the priority pHs that were detected in that individual's wristband. So it's a very dynamic report, and we can run these across hundreds of individuals. So an incredible amount of credit goes to Mike Barton, who is our computer programmer and developed this. From there, we then go back to the same graph description that we had before. What you might not remember, because I went through it quickly, is our graph description was about like 2 inches by 2 inches, and it was crammed next to their actual results. And so what people end up doing is skipping the graph description, going straight to their results, and then saying, wait, I don't know how to read this graph. So we blew it up to an entire page. We added some analogies. We increased sort of the arrows and the descriptions. I should note that this example graph is also dynamic, which mostly is helpful when we have like 2 time points. So if someone only participated in 1 time point, their graph explanation will actually reflect that. And then finally, a couple pages in, we do get to the individual results. These are written at an 8th grade reading level. We've also added a lot of explanation as to why we selected these PAHs to highlight. Once you sort of walk through, like, what did you do? What are PAHs? Where are they found? What's in my wristband? We then ended the report with the same infographic, but mitigation strategies. So if you're concerned, how could you reduce your exposure to PAHs? So I know I'm going through this really quickly. My apologies. I did want to sort of pause and talk about how we actually discussed these individual PAHs, because this is what a lot of our participants are really interested in. So when we worked on this page, we developed several additional criteria for which PAHs we selected. And this came straight from the focus groups. People were like, oh, this is really interesting to learn about naphthalene, but why did you pick this one out of the 62? So originally, we'd selected them based on their relationship to the health outcome of interest, which in this case was respiratory health and asthma. But that wasn't really clear. So we sort of developed 4 criteria that we now follow for most studies. I will say the overarching criteria is that we're only going to look at chemicals that have been detected in over 25% of the study population. Then our 4 criteria are prevalence in the study population. So we often look at the most frequently detected with PAHs that tends to be naphthalene and phenanthrene. From there, we'll also look at chemicals with known health effects, particularly if those match health outcomes of interest to the research team or health outcomes of interest to the study population. So that kind of goes into the third, where we'll look for chemicals that are associated with community or environmental health concerns. For example, naphthalene, yes, it's a respiratory irritant, but I also select it as a chemical of interest when I'm working with communities that are impacted by pollution from creosote plants. Creosote contains quite a bit of naphthalene. Our final criteria, given that we are a Superfund center, we will often select chemicals that are associated with toxic waste sites. And where we get our information to write these little blurbs, showing some of them here, but IARC, National Toxicology Program, ATSDR, PubChem, European Chemical Agency, EPA, CompTox. We will look through all of those sources, and sometimes I have to look through all of them to be able to find enough information to write about these chemicals and put them into a context that's understandable. The other thing we do is we do try and write all of this at an 8th grade reading level. I'm not going to show you how many drafts it took. It takes a lot of writing the same thing differently. Some of the tools we use, though, are the Fleisch Reading Ease and the Fleisch Kincaid grade level. So for reading ease, we're usually trying to get above 60%. You'll see that I did really well on some, less well on others, and we are trying to get underneath an 8th grade reading level. Again, we did pretty well for some, less for others. These are not perfect formulas. There's several others. We've used SMOG, which I love that one mostly because SMOG stands for Simple Measure of Gobbledygook. But there's quite a few other formulas that we're sort of branching out into using to get a more comprehensive understanding of readability. So that is the New York study, but I wanted to pause and put this actually into context. So a lot of our work is around context. But I mentioned that we did focus groups with the participants in New York, and participants were incredibly engaged. I mean, much more engaged than we expected. We held one focus group in English, another in Spanish, and while I don't speak Spanish, I was there to sort of answer sort of the more scientific questions about, what is naphthalene, what is phenanthrene. We had an amazing interpreter, but the participants were actually so engaged, they ended up cutting the interpreter out and just translating amongst themselves. So one woman would ask a question in Spanish, her neighbor would say, oh, here's what she's saying. Can you tell me, why is benzoylpyrene found in cigarette smoke? And so we had this incredible conversation. We actually sort of just stopped the focus group and let participants talk to each other, talk to us, and drive the conversation. And ultimately, probably not surprisingly, someone finally stopped and said, this is really interesting to have all of this data, but like, what does it mean? What is the outcome of all of this? And so we talked about the scientific process, right? And how ultimately we want to get to improved health. But first, we have to figure out what people are even exposed to, right? Like, what pHs are they being exposed to? Then once we know what they're exposed to and what could be contributing those exposures, we can start connecting those exposures to human health. And as people understand that connection, we can then develop interventions, or they can make individual changes and they can improve their health. And so I was like, we're still at this process of figuring out what chemicals you're exposed to. And a woman interrupted me and said, well, you're kind of talking about a staircase, right? Like, we're at the bottom of the stairs, but the data we're helping you generate helps you step up to the next stair. And then as you get more data, you can step up to the next stair, and then you can finally get to the top of that staircase. And so I thought it was just a beautiful analogy. And we really started using this analogy in multiple studies to help set those expectations of, yeah, we are at the bottom of the staircase for a lot of this, but the information that you're helping us generate helps us move to that next stair. So that was New York. Moving into Detroit, we obviously took a lot of the lessons we learned from New York and implemented them in Detroit with the CLEAR study. Like in New York, we had very engaged community liaisons. They helped revise our template. They even helped plan our focus group and identify some of the community concerns. I will flag that one major difference is New York, they got to look at their actual data. In Detroit, we did not use real data for two reasons. One, very pragmatic. We didn't have the data yet. But two, we thought about how we could get the most objective feedback and opinions on the report. So we decided to use mock data. In Detroit, we also had a much more structured focus group. So we used a mixed method approach so we could collect quantitative and qualitative analysis. So that included, sorry, my brain just went blank for a second. Ranking questions to get feedback on how we visualize the data. So using bar charts, using strip charts, using Venn diagrams where possible. Then we used a modified ranking strategy to actually initiate discussion on the layout of the report. I'm not going to spend a lot of time on that, but essentially we gave people a red pen and a black pen, and we said, circle what you like with the black pen, circle what you don't like with the red pen. We can actually get quantitative data on that by adding up how many times people circled different elements, but we found it was a really good discussion tool to go through the focus group and ask people like what worked, what didn't work. This focus group was also held on Zoom because this was in the middle of the COVID-19 pandemic. And so that was another good discussion starter for folks on Zoom. We also used Likert scales, and then we did this in both New York and Detroit, but a thematic analysis of the focus groups. So I'm not going to be able to talk about all of this in the interest of time, but like New York, I wanted to show you the first sort of template that we had. You'll notice this looks very different because this is going to be for 1,500 chemicals. So we tried to take some of those lessons learned. On our cover page, we added more graphics, we had less text, and then on the second page, we had our overall, like, what we found, what we think it means. We had a graph. Again, this was marked as mock data, but just to try and get feedback from people. We also, for the chemical categories, we had 1 page per category. So 1 page for endocrine disruptors, 1 page for PCBs, 1 page for PAHs. So we did show drafts of that to the focus group as well to get their feedback. So after focus group and all of the feedback that we received, what we ended up finding was we still kind of missed the mark in text. They did suggest bullet points if possible, but also to increase the font size. And what we forgot to take from New York was that, yes, people want more graphics, but those take-home messages should be on the very first page. That's what people are interested in. That also, for people that are familiar with the AAAS communication model, it fits that model as well. There was a lot of discussion around color. I'm going to talk about that more in a moment because this brought up some really good accessibility considerations that we've really been moving more towards. We did hit the mark, thankfully, in our language level, in graph literacy, but the question about comparisons came up. And then, interestingly, a conversation around parental guilt came up where our community liaison said, you know, being pregnant, you're focused on so many different things. You're trying to pick, you know, do you use disposable or reusable diapers? What laundry detergent do you have? When do you start solid food? There's so many decisions. And now you're bringing in environmental exposures. Let's make sure that this report doesn't cause parents to feel guilt about exposures that happen during pregnancy and potential health outcomes. So that was something we took really seriously. The final report that we ended up providing, the cover letter doesn't look that much different, but you'll notice that we now have the take-home messages, and we reframed it as community questions and potential answers that we have for that, as well as links to learn more about PAHs. We moved the graphic depicting the 9 different chemical categories onto the second page. This is where our focus group had suggested color coding. So they had said, like, that first circle where it says endocrine disruptors, could you make that green? And then when you go down to the bar chart, that first bar, could that also be green? So you can match, like, oh, endocrine disruptors, here's their level. And then maybe the next one is purple and then red. And so as an organizational scheme, that made a lot of sense, but it wouldn't be very accessible for individuals with color blindness or that struggle with shades and differentiating between color. So I won't go into all the details, but we thought about various different ways that we could use color in an accessible format. And ultimately, it ended up detracting from graph literacy. So we have kept sort of this monochrome version for now, but accessibility is something we're continuing to move forward in to see ways that we can make these maybe more visually appealing without losing the accessibility aspects. In this study, we actually were able to then pull in some comparison data. I mentioned a study we did in Houston. We had then gone back and sampled Houston a year later with a flood, so more baseline. So we were able to compare some of the Detroit results to that baseline in Houston and put that data into context. So that is how we use the focus groups to actually improve the report. Oh, and I've seen a question in the chat. If you don't mind, I'm going to save that for the end because I can talk about that for quite a bit, but that's a great comment. So once we had gone through the report and improved the report based on all that feedback, we actually ended up realizing that beyond just collecting evaluation of like, how do you write at an 8th grade reading level, or do people prefer bar charts or Venn diagrams? We actually saw a lot of the reports building environmental health literacy. So again, I am sure this is a concept everyone is familiar with, but it's the understanding that our environment can impact our health. And beyond that, it's do people understand that connection between their environment and their health? So I'm showing a couple different models here, but this is really based on health literacy. So can you understand something? Can you then scaffold on that to be able to analyze or apply? And then ultimately, can you create? And so there is now a model for environmental health literacy. It's a taxonomy. It was developed by Simafin and Liam O'Fallon at NIEHS. And I'm showing it in the A. So it is structured just like Bloom's taxonomy, where you start with this broad foundation of recognition, and you can build all the way up to create. So in New York, we did this qualitatively and quantitatively. Now, those focus groups, like I said, they were not very structured. It really was more of a community conversation. So we didn't record them, but we did take pretty extensive notes. And so Kylie Riley, who was the PhD student on this project, she then actually ran a thematic analysis of those focus groups along with Kim Burke. And they coded the notes to this EHL taxonomy. And what we actually found is that in New York, all elements of EHL were addressed. So the group actually spent quite a bit of time applying their results, followed by time then, of course, evaluating the report, which is what we asked them to do. Now, they did receive their actual results, so it makes sense they were applying them to their real life. And to give an example of why I think report back can be so impactful, out of this group of 22, one participant had the highest levels of benzoylpyrene. Now, benzoylpyrene is a group 1 carcinogen, so there is no level that is a good level. But she used the table we provided and she tracked over and she's like, oh, so benzoylpyrene is found in cigarette smoke. But she said, but I'm not a smoker, so where could this be coming from? And before I even had a chance to reply, she's like, wait a second, I have a relative that comes over and, you know, she smokes outside, but really she just opens a window and she's smoking in my house. Could that be a possible, you know, place that the benzoylpyrene came from? And of course, we don't know for sure, but we do know it's found in cigarette smoke. And so I said, you know, that is a possibility. Now, it's really interesting. I thought she was going to be upset by having the highest level. She was actually thrilled. And she said, I'm taking this report home. I'm telling my relative they can't smoke in my house anymore. It's bad for our health and it's bad for my baby. And so she was immediately empowered by this, saying like, yeah, I don't like this, but now I know exactly what I can do to address this problem. We also asked people to self-rate their knowledge on if the report increased their knowledge of PAHs. Of the 22 people, only 14 filled out the survey, but 13 of those 14 said that the report added to their knowledge of PAHs on a lot. The other thing we did, because we were concerned about giving back all of this chemical information, and is that going to scare people? And the short answer is yes, but. So a lot of the words that we saw in New York were alarmed, scary. One person, you know, wrote that they freaked out a little bit, but then all of them sort of followed up. And the most pertinent quote that we received was, what you're doing is you're giving information that's never been provided to people before. So that's just inherently valuable. And I will note that we see this across all of our studies, that 90% of participants want their data. And in the studies where we're able to follow up after we give them their data, we say, you know, are you glad that you received your results? And even when people were a little uneasy, they were still glad that they had the information. One of the ways we did this in New York is we did a sentiment analysis. So words that are associated with different emotions are then characterized. The most predominant emotion was fear, but then followed by surprise, and then interestingly, joy and anticipation. So we more broadly lumped those into positive, negative, or neutral emotions and actually saw equivalent numbers of positive and negative. So this is more information for us as researchers to say, like, yes, this information does have benefit to participants. But we do need to be really careful about how we report it back to make sure that it is still beneficial information and not just residing in those emotions of fear. So when we then went to Detroit, this analysis is still ongoing. Taylor Vogel is my evaluator who's done this work. I know I'm getting really close to time, so I'm not going to go through all of this, but we've audio recorded these focus groups and we're able to pull out quotes and saw that even when they're looking at mock data, they're just thinking about how they would apply their results to their life once they get them. And so we actually saw people go through every single level of the environmental health literacy taxonomy, from recognize all the way to create. So with someone recognizing that once they have their results, they might be interested in joining environmental action groups in Detroit or working with other people that are interested in levels of ambient environmental pollution. So across all of this, what we saw is that our reports, whether they had actual data or mock data, because we were just looking to evaluate these, they still have the potential to change behavior. So all groups were concerned about their results. And what we saw mostly is that people found that the results could help them figure out where these chemicals are coming from, but also helped inform mitigation strategy. And then to our surprise, the report back actually led to measurable increases in environmental health literacy, both at the personal level, but as well as at the community level. And all of this is despite limitations where we don't have levels for most of these chemicals. We can't tell you if it's below health guidelines or above health guidelines, but people still found value in their chemical data. So to wrap this up, these are sort of the overall revisions. If I can take those two focus groups that we did, what did we learn? We learned that our reports do have to be cognizant of the time lag, right? We're sampling while people are pregnant, but they're not getting their data until after they've already given birth. In some case, like in New York, it was 5 years later. So we are working on minimizing that time lag as much as possible. We are also very cognizant that we're really kind of reporting for two, right? Because we know that what a parent is exposed to while pregnant, well, those parental exposures can also translate to fetal exposures. And then this was the term our community liaisons use, mom guilt, where we want to make sure that our reports don't make people think that prenatal choices or prenatal scenarios that they don't have control over, right, like industrial air pollution, that can impact long-term child health, but that is not the parent's fault, right? This is information they didn't have, now they have it, and moving forward, what might they want to do with that? So our final next steps, just to let you know where we're taking the outcomes of these focus groups, is to really delve into one, accessibility, but two, comparison data. So how can we start answering this question of how our community compares to other communities? So part of that is just doing more studies to collect more data so that we can compare, but also really thinking about different visualization processes that we can use to, at a glance, say, like, this is what your community looks like, here's how it compares to other communities. So bubble plots is one option, obviously bar charts is another that we're looking into, but I would love to hear from all of you what you might think of would be awesome data visualization that we can do. And with that, I know I talk fast, so apologies, and I'm happy to go over anything else, but just incredible acknowledgements to the very large teams that have worked on this. Obviously, the Anderson Laboratory, we work with Pacific Northwest National Labs, the folks in my group out at New York, Julie Erbsen's team is amazing. I'll shout out Layla Calero. She was our translator. She was able to split between English and Spanish at a rate that I have never seen before. And it was just one of the most incredible liaisons for that project. And then, of course, everyone at the CLEAR Research Group who has been involved in this from the beginning, joined our focus groups, and really helped come up with a lot of the different visualizations. So with that, I'm happy to take questions. I know there's also one in the chat. So Jen, should I just start with that one, or do you want to?
Oh yeah, go ahead. Thank you so much, Diana. It's a fantastic talk. So I'll let you go ahead if you want to start with the chat. And then if others have questions, I don't know if you want to put your hand up or just put them in the chat. I'll try to help where I can if and as needed.
Yeah, so the question was, what type of expertise is important to be included in your research team? So yes, I have someone with computer science expertise and a computer programmer who has been absolutely essential for all of these reports since they are automated and personalized. I don't have a graphic designer on my team, but we do often contract for a graphic designer. That said, most of our infographics are designed using PictoChart. So that is an online software program. I know a lot of people use Canva as well. And how do I pay for all of these talents? In a variety of ways. When possible, we build that into the grant itself as part of our report back process. Through my community engagement core, I've set aside funds to do this. So my community engagement core, we focus on report back a lot. And so we're able to use funds for that. But my computer programmer is on staff. I now do have an evaluator on staff who can help with these focus groups. And then one of my staff has done quite a bit of digging into accessibility, which I mentioned. So can we go beyond an 8th grade reading level? I use software for that as well, but how can we really ensure that our reports are accessible on, for example, a smartphone or color blindness or dyslexia? So it's a pretty interdisciplinary team that we've pulled together over the years. There's another one in the chat, but I wasn't sure if there was anyone that already had their hand up. Right, I can go with the chat. So yeah, a lot of the comments did pertain to individual responsibility. Less so on responsibility of employers or city, state, federal government agencies. Yes, I think that's specific to these studies where we were looking at ambient environmental exposures. I can say working with Superfund, I have done some work where we were looking at like city parks or privately owned properties. In that case, I haven't worked with OSHA, but I have worked with EPA. I've worked with city officials where we can talk less about individual and more like what are the efforts that are being done at a community level or at a city level to try and mitigate exposures. I will say that does change the process in which you report back. I have an infographic I made, it was with the city. That infographic took 1.5 years to make because we were working with so many different parties involved in it, but can be done.
I don't see any other questions in the chat at the moment. So Diana, if you don't mind, I'd like to ask you a question. You made an interesting comment about risk being a feeling and also a quantitative measure. And I was wondering if you could tell us a little bit more about how you kind of bridge the gap between those two different views of risk to, I don't know, try and get at what the community really wants in some of those reports.
I would say the main way we do it in the reports is we never tell people how they should feel about it. Like we never say, don't worry, everything's fine, right? I spend a lot of my time looking at chemical exposure data. And so for me personally, and I have sampled around my house and we're hit by wildfires. And so I've sort of seen these different numbers. I have my own sense of like, this level of naphthalene, I personally am not concerned about, or this level of phenanthrene, I'm not concerned about, but I will never tell someone how they should feel. So we always try and couch it in, here are your results. If it's common levels, we'll say that. We'll say this is something we commonly see at these levels, but we're still going to provide resources so that people can make their own decision if they're comfortable with that or if they're not. I think we also try and include a lot of resources that are at an appropriate context, which has been difficult, to be quite honest. I mean, I think EPA information is fantastic, but those pages often aren't intuitive to a lot of our study participants. So that's why we've moved towards infographics, short videos, that sort of thing to supplement the reports that we have. But short answer, which already is too long, we don't tell people how to think or how to feel about things. We let them have their feels. And if someone comes in and says like, I'm terrified, even though I have the lowest level of phenanthrene, then let's talk about it. Let's let them know where it could be coming from, things that they should look out for and ways to reduce their exposure.
Thank you. I think Paul has his hand up.
Yeah, thank you for that talk. It was fascinating. It's not, I do analytical work. I don't really have community involvement, but it is ultimately where everything needs to end up, that people understand the world we live in. And so my question really is about conveying to people in general that a lot of these exposures, we don't know what they do, and we don't know how much of the modern health issues that we have, the increases in diabetes, the increases in obesity, what's the likelihood that these exposures that we have are really changing how our bodies develop and the diseases, conditions that we're all going to deal with, and that it's part of the world we live in now. I mean, can you convey that to people? And is it clear enough from a scientific viewpoint that we should convey those kind of ideas to people?
Yeah, I mean, I'm going to go beyond just these two studies, but obviously this is something I do quite a bit with. That is the thing that comes out of every study, is participants realizing like, oh, we don't know. Like, we know we're being exposed, but we don't know the impacts of this. And the first thing people say is like, that's really upsetting. That's not always the words they use, but they're like, that, you know, that's unfortunate. They'll sometimes try and couch it that way. But because we've tried to do such a good job of saying that up front and just being honest about the limitations, what usually happens is like, that's really upsetting. But now I see why you have to do these studies so that we can get there. I've seen it done the other way, where people have said like, oh, can you tell me this? And they just get a flat no. And that has not gone over well, because they don't know, is it a no because you don't want to tell me? Is it a no because you think it's too complicated for me? Like, that just opens up this whole sort of like, why are you stonewalling? And it's not a stonewall. It's usually the answer is no because we don't know. But tell people why we don't know. Like, let them know, like, you asked a really good question. That's what we also want to know. That's probably the most we've done it. But yeah, it is incredibly frustrating to say, like, we found all these chemicals and we don't know what it means for the most part. Does that answer your question?
It does. Thanks very much.
Yes. I saw there was share your slides. Yeah, I think I can send those. Jen, should I send them to you? I'll double check with some of my co-authors to make sure that we can share everything.
Yeah, if you send them to me, if it's okay, and then I can help
Mhm.
get them distributed.
Yeah, and then clinical psychologist. Really good question. Yes, so this is the question about how the report back is done in terms of individual one-on-one attention. So we've done it a couple different ways. In New York, people were given their report in advance and then came to the focus group. In New York, we actually also filmed a video. It was myself and one of the graduate students, and we just walked people through the report page by page. That's something we've been interested in possibly doing again. In Detroit, we did not do the one-on-one. We have done offers of like drop-in office hours where we'll be on Zoom for a certain number of hours and people can drop in and ask questions. In a perfect world, I would love to do the one-on-one, but as these studies get larger, you know, when we did our study in Houston, I had over 300 people, and doing the one-on-one was not feasible. So it is kind of this catch-22. We want more people so that we can collect more data, but then we lose the ability to do the one-on-one. So we are trying to just work with these reports to make them hopefully more intuitive. But yeah, it is really a good question of how could we maybe not do one-on-one, but provide more resources. Like, are there video walkthroughs? Are there more drop-in Zoom hours that we can do? Or in some studies, we've actually had the opportunity to do an in-person community sort of conversation afterwards, which has been pretty nice. And then, yeah, if there's someone with them, does it become more like an intervention rather than solely report back? I don't have the ability to answer that as well. I will say I've had people call after their results. I don't know this as much of an intervention, but what's been interesting to me is to see how quickly they take this and they apply it to other things that are going on in their life, where they're like, oh, I didn't think about industrial pollution. Oh, but also there's a landfill down the street. Could that be contributing? And how they just tie it into so many other things going on. But overall, yeah, I think that's a really interesting question. Phone-based apps to provide faster, more individual report back. Do you think this is the future? You should absolutely check out Silent Spring. They have a really cool approach for doing the phone-based apps. I think that's incredible. So I do think it's the future. I don't want to speak for them. I'm not going to. What I do think is, how can we get report back to be automated, one, but also how do we give more of these tools to the first gentleman, Paul, you mentioned you're an analytical, right? Do I expect every analytical chemist or toxicologist to do report back? No, that's simply not feasible. So how can we make tools like these more accessible to people that want to do this without requiring every toxicologist or exposure scientist to also be an expert in community engagement and data visualization and talking about risk and all the different interdisciplinary sciences that go into this. That's what I'm hoping the future is just, sort of like, how do we do this across disciplines? And I think it's possible. I absolutely think it's possible. But bringing in all those disciplines and finding frameworks that people can use this, that is my hope for the future of report back.
All right. Thank you so much, Diana. That was a great, a lot of great questions and good conversation afterward. I think we're about to time here, but I really just want to reiterate, we really appreciate you presenting today and sharing all this with us. It's fantastic and a great learning experience for all of us.
Thank you all for giving up your lunch hour.